Hello all and welcome to this presentation of Cancer Immunotherapy by Bahena and Elena Conroy. As many may know, cancer is a disease where abnormal cells divide out of control and destroy body tissue. Although as humans we have a lot of compensatory to overcome single mutation, cancer overrides this with multiple mutations that allow it to be out of control. It is common to see mutations in a proto-oncogene or tumor suppressor gene, both of which allow the cancerous cells to divide rapidly and survive. Unfortunately, as life goes on, there is a higher probability of accumulating mutations in the body that will result in malignancy. Such mutations cause cells to become cancerous and later detected with tumor-specific or tumor-associated antigens. Although all cancer patients are able to have an adaptive immune response to such cancerous cells, these cells have the ability to escape the immune system. One way for cancer cells to do this is to not express MHC1. By this method, they avoid the recognition by CD8 T cytotoxic cells and are able to continue to proliferate and harm the body. Similarly, as you can see in this figure, they can avoid expressing the MYC receptor in order to prevent natural killer cells from detecting and killing them, as this is the second signal needed for eliminating infected cells. Another way to avoid their destruction deals with the famous cytokines. Rather than releasing cytokines for an inflammation response, cancer cells release specific cytokines such as TGF-beta that suppress inflammation and recruit T regulatory cells. The T regulatory cells then suppress CD8 and CD4 T cells in their role of eliminating infections. With all this being said, treatments that boost the immune system have gained popularity when fighting cancer. One of the methods to do this, and the focus of this project, is immune checkpoint therapy. This method does not kill the cancer cells, rather it prevents the inhibition of immune response that the tumor cells induce.
The method of immune checkpoint inhibitors work by controlling an immune response through T cells. Cell surfaces of T cells have the immune checkpoint receptors. In normal cells, the checkpoints keep T cells inactive to prevent harm of these cells. Cancer uses this to keep T cells off and prevent the death of the cancer cells. The immune checkpoint inhibitors block the receptors and inhibitory action of the T cells, which ultimately allows for T cells to be active and able to attack the cancer cells. The discovery of this immunotherapy was based on CTLA4, an immune checkpoint molecule which competes with CD28 for B7 receptor binding and prevents T cell activation. By blocking CTLA4 with the use of antibodies, the negative regulation of T activation is halted and T cell function can occur. This leads to an anti-tumor response, according to Zadie and Jeffy. Another immune checkpoint pathway that has been blocked is the PD-1 pathway, which is known to decrease the response of T cells in tissues. PD-L1 is a ligand of PD-1 and is associated with inflammatory cytokines and expressed in both hematopoietic and epithelial cells. When using CTLA4 inhibitors alone to fight lung cancer, there are reports that this therapy does not provide a significant effect. When this treatment is combined with chemotherapy, benefits to the patient can be seen in those with non-small cell lung cancer and small cell lung cancer compared to control groups of patients undergoing only chemotherapy. Patients with advanced non-small cell lung cancer that were treated with the PD-1 checkpoint blocking antibodies showed significant results in their overall response, long-term survival, and stability of the disease. When looking at another study reported in non-small cell lung cancer patients, the use of PD-L1 had a 10% response rate and lasted between 12.3 and 16.6 months. There was also a report of a 24% rate using different drug that performs the same method.
More specifically, according to the study with Wang et al., there has been the development and characterization of the IgG4 Nivulum antibody. This PD-1 receptor blocking monoclonal antibody has such high affinity and specificity that it prevents interaction between PD-1 and its ligands. With its inhibition abilities, it is thought to enhance T cell responses and cytokine production. When applied to head and neck squamous cell carcinoma, in a study with Al-Saab et al., Nivulum was seen to have a 7.5 month survival rate compared to 5.1 months from traditional chemotherapy. However, even with such positive results, the inhibition of PD-1 is limited to specific types of cancers as it has an insufficient and heterogeneous expression in tumor microenvironments. Although the CTLA and PD-1 methods are the basis of the methods for cancer checkpoint immunotherapies, other targets have surfaced and are beginning to look more promising. For example, the inhibitory immune checkpoint lymphocyte activation gene 3, also known as LAG3, is another target for cancer immunotherapy. Its promising outcomes started in 2006 and continues to develop in clinical trial. In a study conducted by Long et al., a LAG3 Ig fusion protein displayed promising results for immunotherapy, but its target combination with PD-1 showed even more impressive and efficient clinical results for PD-1 resistant patients. The figure shown displays the LAG3 interaction with MHC2. It inhibits the binding of MHC molecule to a T cell receptor and CD4 through a Kiel motif, ultimately inhibiting the T cell receptor signal. A more recent study conducted by Yu et al. Suggested the use of a specific antibody in order to block the LAG3 checkpoint. This anti-LAG3 antibody is known as LBL-007. Compared to the other antibodies in the study, LBL-007 showed the most significant results for the inhibition of LAG3. It was able to block LAG3 and MHC2 binding as well as its downstream signaling. With this, LBL-007 had become very important to researchers as modifications to its use could lead to even more successful cancer immunotherapies via the LAG3 immune checkpoint. When used against colorectal cancer cells, LBL-007 resulted in a significant delay of tumor growth. With all this being said, LAG3 has grabbed many researchers' attention as it does have an impact on immune response. Therefore, more modifications to such discovery can lead to a more detailed target immunotherapy that stops the negative regulation of T cells.
Based on the studies presented, immune checkpoint therapy has been shown to be a successful treatment for many advanced cancers. In a meta-analysis performed by Nishima et al., the immune checkpoint inhibitor immunotherapy efficiency was looked at based on younger and elder patients and determined that this treatment had improved overall survival compared to control patients in both age groups. Based on this review, the use of immune checkpoint inhibitors has contributed to our understanding to immunotherapies as it can be beneficial for a widespread group of individuals battling advanced cancers. Most importantly, unlike other treatments, this checkpoint targeting is not restricted based on a patient's age. The 2018 Nobel Prize in Physiology of Medicine was awarded for discoveries that led to the care for advanced malignancies through the development of immune checkpoint inhibitors. These immune checkpoint inhibitors have FDA approval in the treatment of non-small cell lung cancer, renal cancer, lymphoma, and a subset of colon cancers, according to Zadie and Jeffy, by extending survival and providing cures for these advanced cancers.
Now that it is known that these can be beneficial, the use of these immune checkpoint molecules in the earlier stages of cancers can be explored. However, the use of such immune checkpoint inhibitors have to continuously be monitored for future research. Constant testing needs to be conducted to ensure that the safety of a patient is not more compromised when already dealing with the cancer. Perhaps future research can target immune checkpoints in groups rather than single targets, as we saw how promising the combination of LAG3 and PD-1 was. Not only can a combination of such checkpoint inhibitors be implemented, such method can be combined with traditional cancer treatments as well. This has the potential to show positive results against tumor growth and the eradication of cancer cells in a shorter period of time. Ultimately, the goal is to continuously investigate these immune checkpoint targets as they can be the future of immuno-oncology. Thank you for listening to this presentation of cancer immunotherapy.